Hello, this is Ameen Ahmed from Near East University. Highest respect to Dr. Muhammad Azuz. So, let's go for our speaks today on the 10 lines. What is CRISPR? So, at the first of the beginning, we're gonna go through some illustration about what is happening in a CRISPR and through the mechanism what is happening inside. A few information about it. So, at the beginning, let's go for the first speech about the CRISPR. So at the starting we have the bacteria mechanism and against the viruses, or we can say in other words what is the bacterial graph when it attacks the viruses, or the same bacteria when its bacterial graph affect and attacks the bacteria. So in general, the bacteria have a virus mechanism to defend against the viruses, such as the bacterial graph, as we mentioned. One key for defenses from the Crispr-Cas system, which allows the bacteria to store the snippets of the viral DNA. So we can see here inside the DNA, viral DNA of the CRISPR in own genome, it's the first same if the same virus attacks again the bacteria, we can use these as the snippets as a templates for to produce the DNA molecules that guide to the Cas proteins. However, to target and destroy the viral DNA, the adaptive immune system helps the bacteria resist in the viral infection. So these are important rules what is happening actually inside the lactic cycle of the bacteria against the bacterial graph inside it. So this is like an elaboration to start with the bacteria mechanism, what is happening inside for the injected DNA and all of these details. For the beginning, let's go for the next mechanism for the Cas1 and Cas2. As we can mention it, we can illustrate more the bacterial Cas1 and 2 plays an important role. Why? We can ask this question. So for the Cas1 and Cas2 inside the CRISPR, the essential components of the CRISPR, because the Cas system involved in adaptation phase where the system captures and incorporates short segments of the foreign DNA. So from the viruses and plasmids. So we can moderate this in the bacterial genome as a spacer Cas2 is also involved in this process. However, the aiding for this operation is integration for this spacer for the Cas1 and Cas2 illustration. For the adaptation for the step is too crucial, it's because it allows the bacterium to remember the genetic material of the past invaders. When the bacterium encounters the same as we call it, the same DNA or similar DNA for the future, the CRISPR system guided by this storage spacer, so can precisely target the enclave, the invading DNA, providing the bacterium with defense mechanism against the specific viruses or other genetic industry. So let's go for more information about what happening inside the Cas1 and Cas2, the crucial defense mechanism inside it for the DNA and RNA, as we see it here into the presentation. So for centrally the adaptation phase and the few steps I can mention it here, since the spacer is creation and the CRISPR DNA biogenesis and interferes. 4 more steps is like the adaptation phase and the expression phase and the interference phase. Also at the last is the memory and specify. So at the beginning, for fast illustration for the Cas1 and Cas2 rule, these proteins works together, okay, during the adaptation. So Cas1 helps in integrating the short DNA sequence spacer from the invading virus or plasmids into the CRISPR array, so in the bacterial genome. So Cas2 is involved to stabilizing the integration of the complex inside this operation. So it's 1-to-1 is connected together for the Cas1 and Cas2. They adaptive together to main the defense bacterial graph mechanism. And for more information is for Cas1 and Cas2 is going to the DNA viral to defend it from it. Okay, so this is like a target enclave Okay, expression and processing have a two transcription and a processing. CRISPR array is transcripted into the long precursor for RNA molecules, while the processing is a procedures like then proceed with a smaller CRISPR RNA as we can see for the CRISPR array. For then proceed into CRISPR RNA, and cRNA as we can illustrate it in the next slide. So units by the enzymes. So however, the interference phase has guided by the cRNA when the bacterium is exposed to the same or similar genetic material. Like the cRNA guides is the Cas proteins such as Cas9 for example is the target DNA sequence, while the Cas proteins acts like a molecular scissors. So the precisely targeting or cleaving invading the DNA or RNA rendering to the infective. For the mirror and specify more illustration about what is happening for the pre-cRNA and after the defending from the virus DNA. The adaptive immunity is the storage spacer in the CRISPR array. They call it the genetic memory, which is of the past infection. So the specificity the system can highly specific targeting the particular sequence based on the storage spacer. While in the accountability, we can say the short of that is the visceral CRISPR has led to the adoption of the powerful gene editing tool, particularly in Cas9 protein for application in virus fields. However, so let's begin with important formation of a CRISPR RNA cRNA. What is happening? These has a few more steps talking about it because it has a transcription and it has an procedure formation of cRNA and processing also mature cRNA. The CRISPR region is bacterial genome containing repetitive of DNA sequence interspersed with a short spacer sequence, which is diverted from the past invaders as we spoke, and transcript. Processor cRNA formation transcript initially long pressure for the molecule containing both for the repetitive and spacer as we can see. So the processing is happening in enzymes within the cell process for the molecule generates the cRNA units. While on the other hand, what processing typically involves the cleaving and processor as a specific sites to release the invading cRNA. Mature cRNA in general we can see after the prokaryotic cell as well the transcript for the DNA repeats interventing the viral DNA fragments as a transcript. So the mature is mature for cRNA consists the spacer sequence as a matches to genetic material of the past invader. The mature cRNA functional molecule that guides the Cas proteins to target DNA or RNA. During the interference phase, what is happening as we can mention it above here for the pre-cRNA inside the CRISPR proteins. cRNA in general serves a molecule guide allowing the CRISPR-Cas system to recognize and target specific sequence when the bacterium incorrects the same similar genetic material in the future. So the targeted recognition of the key aspects is the system adaptive immune system response. Sorry. So let's go for the other slide is produced for the RNA as we mentioned and cRNA in general, which is carries the information about the virus. So this is for the cRNA adaptation because proteins target DNA and RNA during the interference phase as we mentioned. So we can calculate as comes from 4 steps. The 4 steps are transcription, processor, cRNA formation, as well as is the processing and mature cRNA. So after that, for the virus detection, probably as a general, as we can see here at the beginning, the detection and defense mechanism against the viruses and bacteria is involved several sophisticated process or stabilized process. So the first process is the first line immunity as a beginning, because the CRISPR-Cas system, if the bacterium has a CRISPR-Cas system, it can store a short sequence of the foreign DNA spacers as we mentioned in the beginning for illustration for that. So, upon the subsequent of the encounters with the same or similar genetic material, the CRISPR in general Cas system, CRISPR-Cas system in general, the system can use a storage spacer to guide the Cas proteins. So, the precisely target the clave and invader DNA. So, restriction modification systems, which is the bacteria, may restriction the enzymes that recognize the specific RNA, okay? And the DNA, if foreign DNA with this sequence is detected, the enzyme cuts and preventing the virus from replicating. You know, it's like cutting the related sequence between them to not replicating again for this DNA virus. On the other hand, the interferes, we can mention it is with the viral replications, the bacteria. The bacteria can be interfere with the virus stages in the viral replication because of such as a blocking, viral entry, distributing viral transcription, perhaps is or translation, preventing the release of viral particles. For the fifth hand is the toxin antitoxin systems, which is some bacteria have a toxin antitoxin system where the toxin is activated in response to the viral infection, leading to programmed cell death, like it's a recycling and then separated from the viral DNA. The connection between the bacteria in general, as we can mention it, is the communicate and the coordinate defense responses through the quorum sensing, enabling them to collectively through the viral threats what is happening inside the system. So these mechanisms collectively contribute the bacterial immune system against the viruses in general, helping the bacteria for survive and resist the viral infection, which is worth nothing to that. This arms for race between the bacteria and viruses has led to evaluation of diverse defense of the strategies between these parties or particles between them. Let's go for another hand for the CRISPR mediated knockout. So the CRISPR mediated knockout KO, which is we can as a genetic engineering technique that is used for CRISPR-Cas in general system to deactivate the knockout specific gene within organism, the DNA actually. Okay, so simplified overview of the process is generally it has like 5 processes, which is designing the guide RNA and CRISPR Cas9 and treating a specific gene and double strand break and cell repair mechanism. Okay, for as we can see here for the CRISPR mediated knockout is like acts as a molecule, because the scientists designed the scientific RNA molecule called the guide RNA, which is complementary to the target gene DNA. CRISPR-Cas9 is the gRNA combined with the Cas protein. As well, the targeting specific gene is gRNA that directs the CRISPR-Cas to complex specific location in the DNA. Double strand break as well for the Cas9 induced the double strands. For the cell repair mechanism is the cell naturally repair the mechanically inside it. We can go for important 2 rules, which is the knockout effect. If repaired through the same CRISPR-Cas9, which is may repair a process by introduce the interest detection of the gene distributing normal function. The distribution often leads to non-functional or knockout gene. So we mentioned that the CRISPR mediated knockout KO as the phase inside the CRISPR-Cas9, perhaps inside this system what is happening. So mediated knockout widely used to research for study gene function, modules disease, perhaps explore like a potential like therapeutic targets, perhaps modified organisms. So these are all related together. So if we go for a CRISPR-Cas, what's happening for that? We can have a collection. This is the biggest exploring about CRISPR-Cas9 in 2012. Jennifer Doudna and Emmanuel L. and groundbreaking discovery for demonstration that we mentioned, because it has a big evaluation what's made it inside the rule of the CRISPR-Cas in general. So let's go for another part for CRISPR gene editing in animals. But before these some steps, CRISPR-Cas9, Feng Zhong and Jennifer Donna and Emmanuel also with talking about CRISPR gene editing in animal cells in general. These like an evaluation for CRISPR editing in animal cells involving using the CRISPR-Cas system to modify specific gene, and they made a lot of modification, and we can simplify it in a few steps later. It's designing the guide RNA and CRISPR-Cas nucleus, and also deliver into the cells targeting specific gene, as well as DNA cleavage and DNA repair. Gene modification is later made it in the last step to recorrect them again. So, So the CRISPR-Cas9 is like the gene editing in animal cells, which is scientists designed a synthetic RNA molecule that call it guide RNA, gRNA. So it's complementary to target gene DNA sequence. However, so the gRNA combined with the Cas molecules of protein, and then the Cas complex CRISPR-Cas is a complex with along with the gRNA along. So it's delivered into a target animal cells that can be done using the various methods to the viral vectors. So as we can see, there are complementary relationship between them. So other delivery systems like the targeting specific gene is the gRNA guides also as well the CRISPR complex of the precise locations in probably the animal cells, as we can mention it here. So genome to where the target gene is situated. So the Cas9 in DNA cleavage is the break in the DNA at the target cells. So this was what is complementary for it. For DNA repair is the cell natural repair mechanism. It is happening inside the CRISPR-Cas9, which is the modification of it. Analyzes insect selection have 2 points. The scientists, probably as we can, they mention it, analyze the edited genome for the gene modification, as well as the selected cell with intended genetic changes to be for the further research later. CRISPR-Cas gene in general, animal cells have revolution in too many factors, like for disease, improving livestock maybe, and potential therapeutic intervention for genetic disorders. However, so this is a structure involved the CRISPR gene probably in DNA and ribonic acids, the RNA, as well as the nucleotides. As we can see, it's talking about the guanine, cysteine, and thymine, adenine. Perhaps the RNA is only different with the uracil, which is after the DNA, and guanine and cysteine. The difference between the structure of them, both of them on the right and the left drawing. So, perhaps for process for gene editing is what is happening as we mentioned for gene modifications exactly. Alternatively of repair, the template for provide the cells and can be utilized insert or replace of the specific DNA sequence. So, sorry, for the application in animals research, and probably we can go through this some specific areas for the functional NHGEHG, which is this one is the gene modifications. If repaired through, as we can see, the NHEG is the repair process may introduce insertion of the deletion of alleles in the gene distributing in the normal function. So this is the single function and the part of the single function, the modification of the gRNA. So CRISPR gene editing can be applied also on the animals in the clinical, probably in general. So it has like more application animals, creating knockout in animals, perhaps not out functional genomics, educational application, and biomedical and therapeutic uses. So these are the CRISPR gene editing. It can be applied on the animal clinic as the scientists what they reached. So the ethical consideration and challenges, it has a lot of challenges and evaluation probably. But if we went to the creating nick in animals and creating nick out animals, scientists often use CRISPR to generate nick out animals where a specific gene is inactivated or activated or inactivated, so to help to reveal the function of target gene impact organism. Perhaps the nick in is enables the insertion of specific gene for the sequence into the animal's genome. This can be used to introduce to the desired traits, to modify the gene to research purposes. So this is what they mean in the CRISPR for the application on it. Editing widely uses created like animals for modules for studying human disease. However, by introducing the specific gene modifications, researchers can be better to understand the point of the gene in the health and disease for both of them. This is what we can see. The DNA breaks knock out and into the illustration what we spoke in a minute about that, what we have mentioned about the CRISPR for cancer research in all whites for the gene editing, gRNA, and with the CRISPR-9. Thanks for watching. This is an illustration was about the CRISPR and the gene editing. Thank you.